been used to looking at. However, if you identify the appropriate patient population and you apply the appropriate molecularly targeted therapy, in this case Herceptin, the kinds of outcome differences you can see is on the next slide, which is missing, but there is a 50% reduction as opposed to a 2% reduction. So the curves show a dramatic change in outcome. These are the data that were presented at ASCO now 3 years ago. This is the HERA trial. Many investigators in China participated in this trial. It showed the same thing. The hazard ratio decreased to essentially 0.5, so there was a 50% reduction in recurrence as opposed to single-digit percentages. And these are the data which also don't show because of the conversion from Macintosh to PC, but BCRG trial shows essentially the same thing. More than 13,000 women were treated worldwide in 4 major adjuvant trials. For the first time in my memory, every one of those trials showed the identical result. The reduction in recurrences was half, and the improvement in survival has been close to 1/3 at 6 years follow-up. We are waiting for longer follow-up to see that this is sustained, but we are hopeful that this addition has induced an improvement in cure rate. So the lessons from the HER2 story are the identification of the target, validation of the target preclinically, determination of how to best use the antibody preclinically, and then ultimately the translation into initial proof of concept trials which have led to the approval both for metastatic disease and now adjuvant therapy, and we are in the phase of clinical optimization. But the question finally is, can we do even better? And the hope is that further clinical translation of the biologically relevant molecular data will lead to even more effective and less toxic therapy. So we are now using molecular profiling to improve our ability to identify the pathways that are involved that cause these aggressive tumors and to validate new therapeutic approaches. You remember I showed you this slide where there is a dramatic change in the phenotype. It is clear that this one receptor acting by itself cannot do all of this, but instead it needs to engage other pathways and other genes. So while HER2 is an important inciting event, it is the triggering event. It has to engage other genes and pathways to give you such a profound clinical picture and a change in phenotype. If we understand them better, can we develop more better effective therapy? So I will finish on this point by saying we've begun looking at global gene expression profiling of these HER2 positive cells. What you are looking at here is a microarray of RNA expression in HER2 positive cells compared to HER2 positive cells. The cells are genetically identical. A perfect match of the RNA would be a complete fit along the diagonal. And you can see that more than 40,000 genes, 40,000 elements representing 18,000 genes fit very tightly when you compare HER2 positive to HER2 positive cells. However, when you take HER2 positive cells and compare them to HER2 negative cells, where the cells are otherwise genetically identical, this is what you see. You begin to see genes that go up in expression, so each dot represents a separate gene, as well as genes that go down in expression. In the 40,000 elements we looked at, more than 500 changed 2.5-fold, 74 genes increased in expression, 115 decreased in expression, and this is simply a list of the different categories. Other receptors in green, things that go down, in red, things that go up, other growth factors and cytokines. Essentially, there is a large list of the changes of almost 500 elements going up and down. The problem with microarray data is how do you select what is important from this large body of data? So we entered several filters to allow us to try and identify the important genes. We asked for genes that fell into identifiable pathways. We asked for genes that consistently affected multiple cell lines. In other words, every time we introduced HER2, did we see the same change in that gene? Was it consistent across a large cell line panel, as opposed to being unique to only one cell line, which would have made it less interesting? Finally, we know the HER2 phenotype, and where the change is apparent to be likely to contribute to this phenotype. And the last filter was we had an inhibitor of HER2 called Herceptin. So if a gene went up or down and we inhibited the HER2 pathway, would we reverse that change? Would things that went up come back down, and would genes that went down come back up? In asking that question, we found something quite remarkable, although in retrospect, perhaps not surprising. One of the genes that increased significantly in multiple cell lines, from 1.5-fold up to 4.5-fold, is VEGF, the vascular endothelial growth factor, that is part of the angiogenic switch. In addition, angiopoietin, another factor for the Tie receptors, which are important in angiogenesis, and fibroblast growth factor receptor number 4, which also plays a critical role. So it appeared that the HER2 alteration was triggering a change in the VEGF pathway. We wanted to confirm this in multiple cell lines. The next slide shows that consistently, regardless of the cell lines we'd used, this is MCF7, we saw it also in BT20, as well as ER75. Consistently, the protein for VEGF, when we added HER2 to the cells, was dramatically increased in the lysate. The next question was, are these increased VEGF levels associated with an increase in angiogenesis? Are they biologically relevant? And this simply shows you an experiment where we are now looking in the mouse model with human breast cancer cells that do not have the alteration and that do. They are shown here. They are implanted into a window, a glass window, into the subcutaneous tissue of the animal, and we are able then to follow the tumor development, and more importantly, with special filters, we can look at the vascular supply to the tumor. At day 0, we see little differences, but at day 3, here are the HER2 normals, and here are the HER2 positive. Now you see tortuosity of the vessels, you see leakage, you see budding and blebbing, indicating there is neoangiogenesis, or new blood vessel formation that's very active here versus here. By day 7, this is even more apparent. And by day 14, the HER2 normal cells have outgrown their blood supply, and the tumor is undergoing necrosis. But the HER2 positive cells continue to grow, the tumor continues to get quite large, and they are fed by a very rich plexus of vessels. So the VEGF production is playing a role in supplying this tumor with what it needs to grow. Now the final filter, if we add Herceptin and block HER2, will we reduce VEGF levels? And the answer is yes. Here are the control cells. In yellow is VEGF protein production before we add Herceptin. In red is VEGF production after we add Herceptin. There is no change in VEGF production. However, in the HER2 positive cells, the dramatic increase in VEGF production is significantly reduced when we add Herceptin. So an antibody directed against HER2 has a profound effect on VEGF production, indicating that the two pathways are linked. These are all preclinical data. The question is, will we find the same thing in actual clinical specimens? We looked at a total of 611 human breast cancer specimens that were frozen, for which we had long-term follow-up, more than 15-year follow-up.